Alright everybody, welcome back to PLPA 303. Today we're going to be going over Lab 4, finally finishing up with bacteria, and we're going to be moving on to nematology. But before we get into it, I want to talk about I want to give you guys some feedback on your assignments, just some general feedback. Make sure you're reading the questions carefully and that you answer them fully, because some of the questions have two parts to them, and if you only answer one part of the question, then I can only give you half credit. So a lot of you have been losing points on the assignments for not answering the questions fully. Also, when you write down your observations, they have to be descriptive enough to paint a mental image to the reader. For one of the observations of the diseased potato slice, one of you wrote, it was a brown potato and squishy. So it's obviously insufficient, so I didn't give that person any points for that. Same thing with your sketches. Don't just draw lines and circles. I know some of the bacteria are very difficult to observe underneath the microscope, but you got to put a little bit of effort into it. At least write, if you can't do a good sketch, at least write what you see. And then regarding Koch's postulates, on one of the questions for the assignments, it asked what are Koch's postulates, or who was Robert Koch? First of all, Robert Koch did not discover anthrax disease, right? Anthrax disease was around long before Robert Koch was. What Robert Koch did was he discovered the causal agent of anthrax disease, and in doing so he developed a system of identifying which organism causes which disease, and that method is called Koch's postulates. Also, Koch's postulates are not plant pathology specific, although we have adopted them for use in plant pathology. They're in use whenever one organism can be infected by another. So it's plants and animals. Also, some of you were having difficulty between SPP, SSP, and PV for the Latin names of the organisms. So I just wanted to quickly go over them, since I don't think they explain this in your classes. SPP stands for species pluralis, and it's simply all the species within a genus. So for example, we've been looking at Pectobacterium, and there's different types of species within the genus Pectobacterium. So if I were to refer to all of them in writing, I would write Pectobacterium SPP, species pluralis. SSP is subspecies. Subspecies is a taxonomic rank below that of species. So, for example, what we're hoping to isolate, Pectobacterium keratoverum, subspecies keratoverum. This is technically within the species Pectobacterium keratoverum, but because it has specific hosts or specific genes in it that differentiate it from other species, we give it another rank of subspecies. And then PV stands for pathovar. Pathovar will define the host range of the pathogen. So for example, Pseudomonas syringae, pathovar phaseolicola. Phaseolicola means bean, so this is Pseudomonas that infects bean. Any questions on the assignments that you guys have been submitting online? Okay. Alright, so on to the actual lab lecture. So, as I mentioned, we're going to be getting to nematology today. We're going to be doing four things. We're going to be extracting nematode eggs from an infected plant. We're going to be re-inoculating a healthy plant with those eggs, rather than just the soil. We're also going to be taking observations of your McConkey agar plates from last week, and we will be doing step 4 of Koch's postulates. What is step 4 of Koch's postulates? Step 4 is re-isolating the pathogen that we inoculated last week and comparing it to that from step 2. Alright, so let's get into nematodes then. There's different types of nematodes. The ones we're going to be dealing with, though, are called root knot nematodes. As their name implies, they cause knots to form in roots, because that's where they specifically attack, where they start to eat the roots. We're going to be working with Meloidogyne species. So again, that's that SPP, in which you have all the species that fall within this genus Meloidogyne. There's many species described. However, there's only 10 to 15 of them that are of major economic importance, meaning these are the ones that farmers need to watch out for. Collectively, they can attack every crop species because they attack the roots of plants, and all plants have roots. Almost all plants have roots. The exceptions would be your parasitic plants, although they do have a phase in which they have root-like things, but you don't need to know that for this class. All you need to know is that the nematodes attack the roots, so they're soil-dwelling pathogens. And they're most common in warm to temperate tropical climates. They like these climates because nematodes need water in the soil in order to swim around and move around. So nematodes are obligate parasites, meaning they need to reproduce within their host. However, they don't need to stay within their host exclusively. They can survive in the environment for some time, especially when they're in the egg form. So for the nematode itself, I mean, you guys have already seen nematodes in your plant pathology class, but just to quickly go over them, they have this cuticle, which is this flexible exoskeleton that protects them. They've got intestines, they've got a nervous system, reproductive organs, as well as an esophagus. And they're very small. They're typically around 1 millimeter in length. And the smallest objects that your eyes can resolve is about 0.2 millimeters. So you can see them with the naked eye, but just barely. There's also different types of nematodes in terms of their lifestyle. You've got migratory ectoparasites. What does ecto mean? The root ecto. Outside, right. So migratory means it moves around, and ecto means it's going to be, it doesn't burrow into the roots themselves. So these will be on the outside of the root, and their whole life cycle will be outside the root. And they use a stylet, which is kind of like a little syringe that they use to stab the plant cells, where they inject enzymes that break down the cell and kind of digest it for them, and then they use that stylet again to slurp it all up. And so now we'll get into migratory endoparasites. Endo would be the opposite of ecto, so endo would be inside. Migratory endoparasitic nematodes will actually start to move around within the root cells of the plant. So you're going to get a lot of lesions because of that. Think of it like burrowing through the soil. You know, you're moving around all the cells within the root, and that just messes everything up. However, the species of nematode that we're going to be working with has a sedentary endoparasitic lifestyle. Sedentary means it doesn't move around, it kind of, I want to say lazy, but rather just there's not a lot of physical movement. It certainly does do a lot. It also causes galls, so what the nematode does, similar to agrobacterium, is it tricks the plant into growing in that area where the nematode is living in, so that way the nematode has a constant influx of sugars and other nutrients that it needs to grow. So that's whenever we see these on roots, we would call this a knot, right? This just big fat lump. So you got your nematode living in here with its eggs that it excretes, and it just forms a big knot, and that we'll see later in the lab today in person. This is what those knots look like. You can see just kind of lumpy roots, and that's the one of the hallmark symptoms of nematodes. If they're messing with the roots, which absorb water, what type of symptoms would you expect to see in the foliage of the plant? Roots absorb water, right? So what happens if the plant's not getting enough water, or if something's interrupted? Right, it starts to wilt. So you'll see the tomato plants that we have infected here tend to be wilting, and that, you know, not having sufficient water also messes with photosynthesis. So you'll get a lot of chlorosis, a lot of yellowing of the leaves. Now those two, just wilting and chlorosis, are pretty general symptoms. So that's why we mainly look at the roots when it deals with nematodes, because since nematodes are all soil dwelling, they're always going to mess with the roots in one way or another. These are just more slides, more close-up of what that knot looks like in the root. You see you start to get all these kind of lumps and bumps everywhere. It just looks very nasty. You can also get stubby roots, meaning they don't grow as long as they should be. And you can also get excessive branching. That's a result of the stress that the nematode induces on the plant. And sometimes they can just straight up kill the roots as well. Now, within the life cycle of nematodes, you have different stages that we refer to. The stage that we're concerned with is this J2 stage, which is the juvenile 2 stage. It's the most susceptible to chemical treatments. The juvenile 2 stage would be the stage in between which they're in the eggs here and before they infect the root. So they're out in the soil. So why would the J2 stage be so susceptible to chemical treatment? What makes you guys think? You've got your eggs up there in the soil. The eggs are pretty well protective of whatever's growing inside of it, just because it's got a very thick cell wall and enzymes to protect it. But when the juveniles are out in the dirt, you know, they're exposed, they're completely exposed, so any chemicals that you spray into the soil or that you put into the soil are going to affect the J2 stages. However, once those nematodes are actually inside the roots themselves, the root is what protects the nematodes, and then they start to grow into the J3 and J4 stages. So that's why the J2 is very important. Is an important stage in which to target nematodes when you're dealing with this parasite. Now for the controls, I won't go over the controls in detail just because you guys have probably seen it already. But you got exclusion, which is just not planting in the, or not using any material that is known to be infected with nematodes. You've also got avoidance, which means just don't plant in an area that is known to be infested with nematodes. And then there's eradication, in which you actively seek to destroy the nematodes. You've got protection, which is similar to avoidance, except you put up either physical or chemical barriers to prevent nematodes from reaching your plants. And then lastly, you have therapy, in which you use chemicals to treat either the soil or the plant themselves after they've been infected. All right, so that's it for the lecture on nematodes. Pretty simple. Again, we're going to be doing four things. So we're doing 2 parts for the Koch postulates and 2 parts for the nematology. You guys are going to be pairing up, so that way we have enough supplies and time to finish this lab today. All right, let's do Koch's postulates first. So again, step 4, we're going to be re-isolating whatever's growing on your potato, if anything's growing. These slides are very dense. Unfortunately, I don't make these slides, but you guys already know step 2. Step 3, if you recall, that's what we did last week. We inoculated potatoes with our pure culture that we obtained from step 2. So now we should have our diseased potatoes in front of our tables, and we're going to be comparing whatever's growing on our diseased potato to see if it's the same as in step 2. So we got to obtain a pure culture. So you guys are pretty much doing lab 1 today, if you recall what we did there. But by now you guys are experts on using sterile technique and inoculating and plates, streaking as well. So you guys should be able to do that pretty quickly. So what you guys are going to be doing though is you're just going to be putting it on the regular plates that we have here for you, so you can compare them apples to apples. You're also going to be looking at your McConkey agar plates that you streaked on last week. So for that, you're going to be doing observations. Today we have assignments. Let's see, what do we have? Today we have assignments 5 and 6, and parts of 8 and 7. For you online folks, you should be able to finish up parts 8 and 7 today, since we don't give you guys observation problems, but rather things that you have to research on your own. I won't go over the instructions on what to do for the Koch postulates. You're just going to be streaking new plates with using the bacteria that are growing on your potatoes. For the nematology portion, we're going to be extracting the eggs that are growing in the roots from those infected plants that we have in the back. So we'll show them on camera in just a bit. And you'll be isolating those eggs. You'll be looking at them underneath either compound or dissecting microscope, whichever one you choose. And you're going to be taking observations, writing down your observations and sketching them out. So make sure you're detailed enough in that. And then we'll be inoculating the soil of new seedlings of tomato with those eggs. So next week we'll be looking at the nematode itself and see what symptoms we see. Okay, the instructions, you guys have already read the notebook, I hope, so I hope you wrote down what it is you need to do, or that you at least know what you need to do. So that way I don't have to re-explain everything here, but I'll leave this up here while we're doing the actual experiment, and I'll just show you instead in the techniques part of the video how to do today's lab. Let's see. Yeah, so this is the inoculation instructions. Oh, and then I'll show you guys how to use a micropipette that you guys have in front of you, and also how to calculate things. For this lab, you guys don't have to calculate very much. It should be pretty simple. But we'll show you anyways how it's done. And then for you online folks, you have this resource to how to use a micropipette. But we'll be showing you here in lab. So any questions then before we begin? All right. Next, let's do the techniques part.